work on an animal model of addiction, alcohol addiction. And just a few words about alcohol and society and alcohol in our society. And you know that alcohol addiction associated with chronic excessive alcohol consumption is one of the largest health burdens on society. In particular, in Brazil, it is estimated that 59% of the population make use of ethanol in an abusive manner, and approximately 14% of them are dependents. Clear for us, a political health issue. About the alcohol addiction, we can define this pathology, and of course there's many different definitions or description of this pathology, but for our group, we decided to define the alcohol addiction as a complex multifactorial conditions and as defined as a relapsing disorder that is modulated by compulsion to take the drug, the loss of control in limiting intake, and an emergence of a negative emotional state when access to the drug is interrupted. In particular, we are interested in the loss of control of limiting intake. And one of the brain circuits that we are interested in is the mesolimbic dopaminergic system. And we know that this system is implicated in the reinforcing effects of ethanol and also involved in associative learning and reward. And the drugs increase synaptic dopamine concentration in this system. In this picture, we can see a brain, and in the mesolimbic dopaminergic system, drugs act in specific brain regions and integrative systems, like prefrontal cortex, hippocampus, nucleus accumbens, and in particular, the striatum. The striatum is a region of integrating among various neurobiological circuits, like dopaminergic systems, GABAergic system also. And it plays a central role in reward, in the take the decision, the motivated to intake alcohol, for example. And we know that neuroadaptations in this area are linked with the transition of the occasional to the drug habit. And we know that some morphological modifications in this striatum are associated with this drug habit. But we don't know a lot about the molecular regulations in this area and the addicted phenotype. And because of this, we are interested in searching for genes that may be related to the addictive behavior through global transcription analysis in mice exposed to ethanol. And by this strategy, we wanted to find or identify changes in biochemical pathway related to the development and maintenance of ethanol consumption, in particular in the striatum. And our methods, the first question was, do we have an animal model of addiction, ethanol addiction? In fact, we developed an animal model based on a three-bottle, free-choice paradigm. For doing it, we had 80 outbred strain male mice, individually housed in a normal conditions, environmental conditions, and our protocol had 4 different phases. The first one, that had 2 weeks, we housed 60 animals, had access of different bottles of different solutions, one of water, one of ethanol 5%, and another ethanol 10%. 20 mice had access only to water and remained only with water all the experimental long. At the end of the 10 weeks, we removed the ethanol solutions and named withdrawal phase and the abstinence phase, for example. And after the 2 weeks, we introduced the ethanol solutions during 2 weeks, for more 2 weeks. And the last 2 weeks, we mixed the quinine in this alcohol ethanol solutions, an untestable solution. And at the end of the protocol, we could classify our animals. But during the animal experiment, we made some records. For example, we measured the fluid intake every 2 days, and animals were weighed once a week. We changed the position of the bottles every 2 days. We made some behavior tests at the end of each phase, and at the end, we determined the blood ethanol concentration for each animal. And according to their alcohol intake, we could classify the animals in 3 different groups. The first group, and the second one, by the way, had ethanol preference. The first one had ethanol preference in a constant intake, even in the adulteration phase. And the back was the highest back that we could find. In fact, this concentration of blood ethanol is considered an intoxication level for the mouse organism. The second way, and the addicted animal, is this. The second group, the heavy animal, had ethanol preference, but in the adulteration phase, they changed. They preferred clear water than ethanol. And the back was lowest than the first one. The third group, the light animal, this animal had water preference all protocol long. And the back was the lowest back found in our animals. And of course, the control animal here. Then, the animals were euthanized, and the striatum were extracted, and we made our global study expression genes by the gene array strategy, and for it we used 4 animals for the light group against 4 animals for the addicted group, the 2 extreme groups that had access to ethanol. And we used the array by affirmatics, and we made some huge bioinformatic analysis, and by hang prod, we could find 126 genes that were different expressed or regulated between these 2 groups of animals. Okay? With these genes, we decided then to study if we had a particular pathway involved in this kind of phenotype. And by it, we used the Metacore tool to try to understand the pathway. And by our results, we found some pathways, and some pathways involved in addiction phenotype or alcohol intake, like this pathway and regulated an D1A receptor of dopamine, or this pathway controlling the receptor of kappa-type opioid. But one of them, the most important, based on the p-value, the lowest p-value found, was a new pathway named LRRK2, or in neurons in Parkinson's disease. In this pathway has 33 genes, and in our groups, in our animals, we found 10 of 33 genes, different regulated in our 2 groups of animals. Okay? What is this pathway? What is the importance of this pathway? This pathway, of course, the name, is previously related to the Parkinson's disease, and the main gene, or the main protein, is the name of the pathway, and it's a leucine which repeat in kinase 2, or Dardarin kinase, and is involved with dendritic spine formation in its striatal neurons, and also with uptake of protein synapses compounds. And this picture, I showed just a schema of this pathway, and here we have the synaptic gap, and after the release of the neurotransmitter in the synaptic gap, the vesicle and its components and membrane components are endocytosed and transported to the endosome, where it will be reloaded with a new neurotransmitter molecules and reused in the next action potential. And the genes and the proteins of this pathway are related here to the structure, function, and mobility action of the endocytosis process. Okay? One of the protein that is endocytosed in this pathway is the dopamine transporter protein. And because of this, and considering, one, the endocytose plays a key role in recycling DHT by presynaptic neuron, the DHT controls the availability of dopamine in the synaptic gap, and the dopaminergic tone, or the availability of dopamine, is directly related to the genesis of the addictions. We decided to understand a little bit more about this pathway in our animals. And by it, we decided to investigate it, if this pathway could be involved in addictive behavior, evidence in our animals models. And we decided to select 9 genes of this pathway pre-detected in our microarray strategy and validated each one by real time, now using the 4 groups of animals. And here I show you one of our results, and here we have 3 genes, and an actin gene, a clathrin gene, and a tubulin gene. This gene codify, all these genes codify a structural protein, and they are related to each other, and the function of this is to maintain the structure, the cytoskeleton of the endocytes. Okay? And all of these genes, or the mRNA, are down-regulated in all our 3 groups of animals that had access to ethanol when compared to the control group. This one, and this one. And more than, this gene, the main gene in this pathway, the LRRK2 gene, is upregulated only in the addicted group. And the other groups, these genes are downregulated. What is this gene? This gene codifies a protein that plays a key role on integrative and connective of the motility function of this pathway with the striatal neurons, or the plasticity of the striatal neurons. Okay? And here in our addicted animals, it is upregulated. And in conclusion, our results suggest for the first time that the involvement of the LRRK2 pathway over the uncontrolled behavior seen in the alcohol use disorders. And the overexpression of the main gene of this pathway and possibility its protein in the striatal neurons promote an imbalance of those processes, promoting then an impairment in the dopaminergic system and potentially contributing for the loss of control of alcohol intake. By our perspective, we can make some future functional experiments are needed in order to correlate the RNA alterations with the protein levels, and efforts should be made to see the direct relationship between this gene and the alcohol intake behavior through in vivo genetic manipulations like RNA or transgenic animals. And I would like to thank our collaborators from different kind of institutions in my country, for example, the University of Paraná, the University of São Paulo, and the University of Campinas. And a huge thank you for my group, my students, who's work hard in our lab every day to make these results inside and give me a chance to show you our work. And thank you for the organizers of the conference. Thank you very much.
Thank you, Dr. Anna.